Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa, where we take difficult biological concepts and make them easy to understand. Every minute, your kidneys filter about 120 to 125 milliliters of blood plasma. That's roughly 50 gallons per day. But how does this complex process work? In this video, we'll break down renal physiology, starting with glomerular filtration, then renal autoregulation, tubular reabsorption, and tubular secretion. By the end, you'll have a clear understanding of how your kidneys maintain balance, regulate blood pressure, and remove waste. The first step in urine formation is glomerular filtration. This occurs in the renal corpuscle, specifically inside the glomerulus. The fluid that enters into the capsular space is called glomerular filtrate. More than 99% of the filtrate returns to the blood via tubular reabsorption, so only about 1 to 2 liters is actually excreted as urine. The filtration membrane is a 3-layered barrier that determines what actually gets filtered. It is composed of fenestrated capillary endothelium. Remember, fenestrated capillaries have large pores that allow most solutes to pass, but it blocks those large proteins and blood cells, keeping them inside the capillaries. Located between the capillaries of the glomerulus and between afferent and efferent arterioles are mesangial cells. These contractile cells can regulate blood flow by contracting or relaxing, playing a crucial role in maintaining glomerular function. And then we also have a basement membrane. This negatively charged layer repels large proteins like albumin to prevent them from entering the filtrate, but allow water and most solutes to pass through. And finally, there are podocytes. These specialized cells in the Bowman's capsule have foot-like processes called pedicels. These wrap around the glomerular capillaries and form filtration slits to further control filtration selectively. Only molecules smaller than 7 nanometers can pass, ensuring that only water, glucose, ions, amino acids, and waste products such as urea and ammonia enter the filtrate. Glomerular filtration depends on pressure differences across the filtration membrane. The driving force behind this is called net filtration pressure, which is the balance of 3 forces: 1 that promotes filtration and 2 that oppose filtration. We have glomerular blood hydrostatic pressure, which is the pushing force that is exerted by blood pressure in the glomerular capillaries. This pressure promotes filtration by forcing water and solutes through the filtration membrane, and it's about 55 millimeters of mercury. We also have capsular hydrostatic pressure. This pressure opposes filtration and is the pressure that is exerted from the fluid that is already inside Bowman's capsule. It is about 15 millimeters of mercury. And then there's blood colloid osmotic pressure. This is the pulling force created by plasma proteins in the blood. This pressure also opposes filtration and is about 30 millimeters of mercury. Net filtration pressure is the total pressure that promotes filtration and is determined by the following equation, where net filtration pressure equals glomerular blood hydrostatic pressure minus capsular hydrostatic pressure plus the blood colloid osmotic pressure. And it should be around 10 millimeters of mercury. This favors filtration, ensuring plasma is efficiently filtered into the nephron. Glomerular filtration rate is the amount of filtrate produced by all renal corpuscles in both kidneys per minute. Normally, this is about 125 milliliters per minute in males and 105 milliliters per minute in females. Homeostasis of body fluids requires that the kidneys maintain a relatively constant glomerular filtration rate. It is vital because if it is too high, important substances like water and ions won't be reabsorbed, which means that they get lost in the urine. If it's too low, then waste products aren't filtered efficiently, leading to toxicity. There are three mechanisms that control glomerular filtration rate: renal autoregulation, neural regulation, and hormonal control. These mechanisms work by adjusting the afferent arteriole, the small artery that supplies blood to the nephron. When the afferent arteriole constricts, less blood reaches the glomerulus. This reduces filtration pressure and slows down urine formation. On the other hand, when the afferent arteriole dilates, more blood flows in, increasing filtration pressure and boosting filtration rate. The kidneys themselves are able to self-regulate and maintain a constant blood flow through the glomerulus. Your kidneys self-regulate glomerular filtration rate through two key mechanisms. The first is the myogenic mechanism. This mechanism works by signals to the smooth muscle in the afferent arteriole wall. When blood pressure rises, the smooth muscle stretches, triggering contraction and constriction of the blood vessel. If blood pressure drops, the smooth muscle fibers are stretched less, and so the muscle relaxes. If blood pressure rises, the afferent arteriole constricts to reduce glomerular filtration rate. This decreases blood volume in the glomerulus and brings glomerular filtration rate back to normal. If blood pressure drops, then the afferent arteriole will dilate to increase glomerular filtration rate. This increases blood flow into the glomerulus and increases glomerular filtration rate. The other mechanism is tubuloglomerular feedback. If glomerular filtration rate is too high, then the filtrate is going to flow through the nephron too quickly, and there is going to be little time for sodium reabsorption. If glomerular filtration rate is too low, then filtrate is going to flow through slowly, and there is going to be lots of time for sodium reabsorption. The macula densa that is found in the ascending limb of the loop of Henle that we talked about in the last video detects sodium levels and adjusts the afferent arteriole diameter to maintain stable filtration. This feedback works more slowly than the myogenic mechanism. There is also neural regulation of glomerular filtration rate. This includes the sympathetic nervous system. Autonomic nervous system fibers innervate kidney blood vessels. These release norepinephrine, causing vasoconstriction. This constricts the afferent arterioles during stress, which reduces glomerular filtration rate. Reducing the blood flow in the kidney allows the body to reduce urine output, which conserves blood volume. It also allows greater blood flow to other body tissues. This is important when the sympathetic system is kicked on during times of exercise, stress, or emergency situations. In these moments, the body prioritizes sending blood to essential organs like the brain, heart, and muscles, ensuring that they receive enough oxygen and nutrients to handle the increased demand. This response, often called the fight or flight reaction, helps the body maintain blood pressure, improve oxygen delivery, and enhance physical performance when faced with challenges like intense physical activity, injury, or danger. And finally, there is the hormonal regulation of glomerular filtration rate. Two hormones that contribute to this are angiotensin II and atrial natriuretic peptide. Angiotensin II is a very potent vasoconstrictor that can constrict both the afferent and efferent arterioles, reducing blood flow in the glomerulus, therefore reducing glomerular filtration rate. An atrial natriuretic peptide is released by the cells of the right atrium in response to increased blood pressure to the heart. This hormone causes the glomerular mesangial cells to relax, increasing the surface area available for filtration. This then increases glomerular filtration rate, helping eliminate excess fluid. Reabsorption is the return of the filtered fluid from the nephron back to the bloodstream. About 99% of the filtered water is reabsorbed, and the majority of that reabsorption occurs in the proximal convoluted tubule. After the filtrate moves through the proximal convoluted tubule, the cells along the rest of the nephron adjust reabsorption to keep water and key ions in balance, ensuring that the body maintains homeostasis. Reabsorption occurs through two pathways. The first is the paracellular route. This is where substances pass between the cells via tight junctions. The tight junctions in the nephron do not completely seal off the interstitial fluid from the tubule fluid. They are leaky, allowing substances to pass between cells. This is a passive process. And then there's transcellular route, and this is where the substances pass through the cells via transport proteins. This process can be passive or active. There are also some common transport mechanisms that we're going to see throughout the nephron. The first is primary active transport. This process requires energy in the form of ATP, and an example of this that we'll see are sodium-potassium pumps. And then there's secondary active transport. This is where symporters move two or more substances in the same direction across the membrane. For example, in the proximal convoluted tubule, the sodium glucose symporter allows sodium and glucose to enter the cell together, enabling glucose reabsorption. There are also antiporters, which transport substances in opposite directions. A key example is the sodium hydrogen antiporter, or the sodium hydrogen exchanger. This is found in the proximal convoluted tubule, and it reabsorbs sodium while secreting hydrogen ions, helping to regulate pH balance. These processes depend on the energy provided by primary active transport mechanisms. Another way that we have transport is through obligatory water reabsorption. The majority of water is reabsorbed along with solutes like sodium, chloride, and glucose, and so this is so named because water is obliged to follow by osmosis as the solutes are reabsorbed, right? Trying to reach that homeostasis. So the solutes get reabsorbed, and then water is obliged to follow to maintain that balance. This occurs in the proximal convoluted tubule and the descending limb of the nephron, as in these areas, the cells are permeable to water. And then there's facultative water reabsorption. This water reabsorption is regulated by antidiuretic hormone and occurs in the late distal convoluted tubule and collecting ducts. The majority of reabsorption, about 65%, occurs in the proximal convoluted tubule. This includes 65% of water, sodium, potassium, and calcium, 100% of organic solutes such as glucose and amino acids, 50% of chloride, and 80% bicarbonate. The proximal convoluted tubule also secretes hydrogen ions and toxins such as ammonium ions and urea. Most solute reabsorption in the proximal convoluted tubule involves sodium. We see this with sodium symporters, and these transport sodium along with important nutrients like amino acids and phosphate, ensuring that they're reabsorbed instead of lost in the urine, so bringing that back into the body. The sodium glucose symporter. This transporter pulls sodium and glucose together into the cells, preventing glucose loss. So even though glucose can enter into the nephron, it is all pulled out in the proximal convoluted tubule and put back into the blood. However, in cases like diabetes where blood glucose levels get too high, it can spill into the urine, stay in the urine, where it stays in the nephron and then it goes into the urine and then would be detected within the urine. And then there's the sodium hydrogen antiporter. This one's a pH balancer. It reabsorbs sodium while secreting hydrogen ions, helping to regulate acid-base balance and reclaim bicarbonate. The cells of the proximal convoluted tubule and the descending limb of the loop of Henle are very permeable to water due to aquaporin 1. These proteins create a water channel that allows water to be easily reabsorbed. Now we move to the loop of Henle. At this point, the tubular fluid's chemical composition is very different from the original filtrate at the glomerulus, since glucose, amino acids, and other key nutrients have already been reabsorbed. However, despite these changes, osmolarity remains similar to blood because water reabsorption has followed solute reabsorption throughout the proximal convoluted tubule. The nephron loop plays a crucial role in further concentrating and diluting the filtrate. It reabsorbs about 15% of the filtered water, 25% of the filtered sodium, calcium, and potassium, 35% of the filtered chloride, and 10% of filtered bicarbonate. It also reabsorbs small amounts of magnesium. Here, for the first time, water reabsorption is no longer automatically linked to solute reabsorption. This separation is key to the kidney's ability to concentrate or dilute urine. The descending limb of the loop of Henle is highly permeable to water, but not solutes. As water exits, the filtrate becomes more concentrated, and since only water can be reabsorbed here, this increases the osmolarity of the fluid as it moves down the tube. In the ascending limb of the loop of Henle, we see that these cells are impermeable to water. So the thick ascending limb contains these sodium, potassium, and chloride symporters that function in sodium and chloride reabsorption. And since this ascending limb is impermeable to water, and then little or no water can be reabsorbed as the filtrate moves through the ascending limb, and those solutes are reabsorbed but not the water, this will decrease the osmolarity of the fluid as it moves up the tube. In the early distal convoluted tubule, about 5% of the filtered sodium and chloride is reabsorbed through sodium chloride symporters. This segment is also a key site where parathyroid hormone regulates calcium reabsorption, adjusting based on the body's needs. By the time the fluid reaches the end of the distal convoluted tubule, 90 to 95% of filtered solutes and fluids have been reabsorbed and returned to the bloodstream. Two key cell types play a role in this final regulation. The principal cells reabsorb sodium and secrete potassium. These cells also have receptors for the hormones aldosterone and antidiuretic hormone, which regulates sodium and water balance. The intercalated cells help regulate blood pH by reabsorbing bicarbonate and secreting hydrogen ions into the filtrate. These cells also reabsorb potassium. At this point, the amount of water reabsorption and solute secretion depends on the body's needs, and for the most part is regulated by hormones. 5 key hormones regulate the reabsorption of sodium, calcium, and water in the late distal convoluted tubule and collecting duct, as well as potassium secretion. These include angiotensin II, aldosterone, antidiuretic hormone, atrial natriuretic peptide, and parathyroid hormone. Let's first talk about the renin-angiotensin-aldosterone system, abbreviated RAS. When blood pressure drops, the kidneys release the enzyme renin from the juxtaglomerular cells into the blood. This triggers a cascade where renin cleaves angiotensinogen, which is synthesized by hepatocytes, liver cells, into angiotensin I. Another enzyme, angiotensin converting enzyme, abbreviated ACE, then converts angiotensin I to angiotensin II. Angiotensin II can then cause vasoconstriction of the afferent arterioles, which decreases glomerular filtration rate. It also increases the reabsorption of sodium, chloride, and water in the proximal convoluted tubule by stimulating the activity of sodium hydrogen antiporters. And it stimulates aldosterone release from the adrenal cortex. Aldosterone then increases sodium and water reabsorption while secreting potassium in the distal tubule and the collecting duct, helping to raise blood pressure and blood volume. Antidiuretic hormone, also known as vasopressin, is secreted from the posterior pituitary and plays a role in water balance. This hormone increases water permeability in the late distal convoluted tubule and collecting duct by inserting aquaporin 2 channels. This allows for greater water reabsorption. This leads to an increase in blood volume and blood pressure. The amount of antidiuretic hormone release depends on the body's hydration status, whether it is hydrated, dehydrated, or overhydrated. Atrial natriuretic peptide is released by the cells of the right atrium when there is a large increase in blood volume to the heart. This hormone reduces blood pressure by inhibiting the reabsorption of sodium and water in the proximal convoluted tubule and collecting duct. It also suppresses the secretion of aldosterone and antidiuretic hormone, leading to increased sodium and water excretion in the urine. This overall effect lowers blood volume and blood pressure, helping to prevent excessive fluid retention. While the previous hormones regulate water balance, parathyroid hormone is essential for calcium homeostasis. When blood calcium levels drop, the parathyroid glands are stimulated to release parathyroid hormone. This hormone then stimulates the cells in the early distal convoluted tubule to increase calcium reabsorption, raising blood calcium levels. It also inhibits phosphate reabsorption in the proximal convoluted tubules, leaving phosphate in the filtrate to be excreted in the urine. Your kidneys are truly remarkable filtration systems, working around the clock to maintain balance in your body. This process begins in the renal corpuscle, where glomerular filtration starts urine formation. From there, the glomerular filtration rate regulation plays a crucial role in maintaining stability, while tubular reabsorption and secretion precisely control what stays and what leaves. And let's not forget the hormones that fine-tune kidney function, adjusting water, electrolytes, and blood pressure to keep everything in balance. If you found this video helpful, don't forget to like, share, and subscribe so that you never miss out on new content. In our next video, we'll continue our journey through kidney physiology and explore how dilute and concentrated urine are formed. Stay tuned, and I'll see you in the next video.